अभी.
ओके क्या जब नोट चाटेगा तब बताना.
प्लीज.
मैं बात कर रहा था. ये क्या है?
Infinite mirror.
What the heck?
Screenshot करने से.
Yes, take a screenshot. Please take a screenshot. Please.
Huh?
Take a screenshot of it.
Screenshot.
Yes. என்ன வந்துதா? அதெல்லாம் நேத்து
बस यहाँ पे थोड़ा
ये वाला ऑन है.
नहीं ये करो कर. मैं इसीलिए अभी बता रहा हूँ.
क्योंकि मेरे को जब इसमें सेशन में रहेगा रूम मोड तो
हाँ.
बस 5 से 10 मिनट के लिए मैं बंद रखूंगा.
कोई
वो वाला ऑन रहेगा.
जितना बंद कर दो. Hi, good evening. Hello, I'm audible? Hello.
Yes, sir.
Yeah, so others are joining. Any update on that?
That's not there.
I mean, others are joining. Do you know any update regarding this?
No, sir.
Okay, fine.
Actually, sir, I have also some work. मेरा exam है actually Monday को.
Okay, okay.
So can I leave or?
Yeah, it's your call actually.
Okay, sir.
Okay. Okay, so good evening. So in this session we will discuss about some immunological tools and techniques. So in previous sessions we discussed about B cell activation, B cell maturation, and when and how B cells get differentiated into plasma B cell and memory B cell specifically, and also we discuss about the T cells, how T cells when it uh, recognize antigen peptide which is presented by MHC, and how it gets activated, and how in turn it activates the B cell so that they can produce the antibody and all. So that we have seen in the previous sessions, and also we covered an interesting thing like how TCR and B cell receptor, how they have the ability of showing variance regarding um, different antigens. So to generate this broad spectrum of uh, antibody pool, or a broad spectrum of the T cell pool, you need variation in their variable region of TCR and variable region of the antibody. So those things are for T cell. The TCR variation is achieved by the VDJ, by recombination. So and for B cell which produces the antibody, for the variable region of the antibody, this uh, pool of the diverse antibody is achieved by somatic hypermutation, and which if we study biochemically, that is achieved by VDJ recombination for heavy chain and VJ recombination for light chain. And also you can get different classes of antibodies, like for example IgG, IgM, IgD, IgA, and all. For that, this particular variable region of the antibody can decide which class to choose. For example, if it will choose the C mu, which is a constant region, then that antibody will so that gene will encode IgM. So that's how they can switch the classes between the antibodies, and they can provide uh, diverse pool of antibody with different variable region and different classes. So that's what we studied in our last few sessions. So in this session we will study the tools and techniques which is majorly used for determining the concentration of antigen and antibody, and to find the concentration of antibody titer in a blood serum when the patient is infected with some antigen. So those things, which techniques are used, so those things will be discussed here. Also we will discuss how can we sort the cells. Okay, uh, can you hear me? Hello, am I audible? Okay, so in this session we will discuss about the tools and techniques for immunology, which is used to determine the concentration of antigen antibody and determining concentration of a specific antibody in a patient blood serum which is infected by pathogen. So those techniques will be discussed here. So all of these techniques are based on a very common, biological phenomenon, which is antigen antibody interaction. The part of antigen which interacts with the antibody is known as epitope, and the part of antibody which interacts with antigen is paratope. So as we discussed in the last few classes, that has uh, epitope generally resides in the variable region of a antibody, and this variable region contains three CDR regions, CDR1, CDR2 and CDR3, which shows the highest variance and the corresponding amino acids on those positions are extremely not conserved. That's why it can, those are the places where you can maximum variance of antibody, which will actually create a broad spectrum of diverse pool of antibody which has different variable region. So those are the CDI region, which is of course the part of the antigenic epitope. So antigen can make interaction with antibody by some bonds. For example, hydrogen bond. Here you can see in this figure, the lysine has made a hydrogen bond with OH group of serine. So here the carbonyl oxygen, this carbonyl oxygen of this lysine amino acid residue has make a hydrogen bond with hydrogen atom of this OH group of this serine amino acid. Another example is ionic bond. For example, here you can see that the O minus atom of glutamic acid, it makes a ionic bond with the opposite charge of NH3 plus. So that's how ionic bond can be made between antibody and antigens. And there were some other interactions, for example hydrophobic interactions, where two hydrophobic groups come to close proximity, then they make interaction which is known as hydrophobic interaction. Another thing is van der Waals interaction and ionic bond. So this fourth kind of interaction we can see, all of them are of course non-covalent: hydrogen bond, ionic bond, hydrophobic interaction and van der Waals. All of them are non-covalent interaction which is found between antigen and antibody. So now in this diagram you can see that uh, they they make two compartment. One is denoted as A, another one is denoted as B. So so at the initial stage only B compartment has antigen, but when it will allow to flow of this antigen through this semi-permeable membrane, then you can see that after some times when it reaches its equilibrium, some of the B molecules has traveled to compartment A, right? So upon equilibrium, you can see that the concentration of A will be increased, because initially it was 0, but when you will allow the migration of antigen from B to A through the semi-permeable membrane, then the A compartment will start rising in terms of concentration of this antigen. So that's why this graph is increasing here according throughout the time, and the exactly opposite thing will be happen for compartment B, because in compartment B initially there was all of the antigen was in compartment B, but after this migration, when it reaches to the equilibrium, then the compartment B will start losing the antigen, right? So initially antigen so initially compartment B had all of the antigen, so that's why it is showing 100%, and compartment A had nothing, so it is showing 0%. But after time goes, the compartment A will start being enriched with antigen, and compartment B will lose antigen. So at the point of equilibrium, the concentration of antigen in compartment B will be equal to the concentration of antigen in compartment A. So now we can see that the this system has reached its equilibrium because now A and B both compartment has equal concentration of the antigen. So this is your control where no antibody was added. Now imagine one situation in your experiment. You have added antibody in A compartment. So antigens are in B compartment and antibody are in A compartment, initial state. After some times, when you will allow this antigen B to migrate through the semipermeable membrane to compartment A, then what will happen? Some of this antigen which which has been traveled from B to A, they will be bound by this, or yeah. So they will be bound by the antibodies which are present in compartment A, right? So and one thing I forgot to mention, that this uh, antigenic fragments, they are radio labeled. Okay, so you can easily measure their intensity, or you can measure their concentration by the radioactive intensity, and that's how this all this graph has been plotted. So when you put an antibody in this compartment A, then what will happen? Antigens from compartment B will start migrating to compartment A, and some of them will be bound, right? So if you see the graph, then of course initially compartment B had 100%, compartment A had 0% antigen. Now, when the time when time goes, then antigen A will start increasing, and the antigen will decreasing in compartment B because some of them will migrate to A, right? But the problem is here it will not get the at the equilibrium stage, unlike here where concentration of antigen in A and B compartment was equal, but here that stage will not reached. Rather, in equilibrium there will be difference between the antigen concentration in compartment A and compartment B. So that difference is known as the concentration of the ligand which is bound by antibody. Here ligand is nothing but antigen. So this difference is the amount of antigen that is bound by antibody. Why? Why so? Why they didn't become equal here? Because when the antigen started migrating from B to A, then some of them was entrapped by antigen, right? So they are not free. So the flow will be continued until the both of the compartment A and compartment B will have equal amount of free antigen, right? So that's why in the control experiment, if there was 10 antigen peptide, then out of them 5 would go to A and 5 would reside in the B. So 5 in A, 5 in B, equilibrium. So that's why they are equal in concentration. But for example here, if there was initially 10, then if 5 goes to compartment A, then suppose 3 of them will be entrapped by the antibody, right? So now 3 of them are not available, they are not free, only 2 are left which are free in compartment A, right? So in order to so now if it wants to attain the equilibrium in compartment B, then compartment B has to release more antigenic peptide to A, so that the number will be 2 here, right? So that's why now if you will calculate the total antigen in compartment A at the equilibrium, then the total antigen concentration at the equilibrium in compartment A will be more than the antigen concentration is compartment B. Although the equilibrium has been reached, but equilibrium will be reached based upon the availability of free antigen. If the free antigens are equal in both of these compartments, then equilibrium will be attained. But because of the presence of the antibody along with some free antigen, the compartment A will also have some antibody bound antigen. So the total will be more in compartment A than compartment B. So that's why this difference will be there between compartment A and compartment B, and this difference is the concentration of the antigen which is bound by antibody. So that is experiment to show this antigen antibody interaction and how to measure the antigens which are bound to the antibody. So moving on to next, uh, yeah. So next, the precipitation reaction. So what is precipitation? Precipitation is when antigen and antibody, when they bind, and if they make a cross link. Cross link means if if there will be a mesh, there will be a network between the antigen antibody complex. If that is so, then this will make a uh, insoluble precipitation, that is called precipitation reaction. So for example, if we take the monoclonal antibody, suppose your reaction mixture has a monoclonal antibody, then whatever antigen you will give, it will only, these antibodies will only bind to that one particular epitope. For example, these blue antibodies will bind to the blue epitope because they are monoclonal antibody, right? So that's why, as the antibody can only bind to a particular epitope in the antigen, then it will lose the ability of uh, binding to multiple antigens, or making this uh, cross link or precipitation, right? But in the case of polyclonal antiserum has polyclonal antibody. Now polyclonal antibody can bind to more than one epitope. So this red one, it is binding here, the blue one is binding the blue epitope, right? So that's how they can make a network or cross link, which will give you a insoluble precipitation, right? So that's why this polyclonal antiserum can make this precipitation, right? But the monoclonal antibody cannot make this uh, precipitation. So this precipitation reaction is, this is very important, that the precipitation reaction is only be successful, can only be done with polyclonal antiserum, not with monoclonal antibody. So now if you will see the precipitation curve, then initially what was happened, so in the x-axis there is antigen added, antigen concentration, and y-axis is antibody concentration. So initially there was antibody excess zone, where you have just start adding the antigen. Initially there was no antigen, and then you gradually started adding the antigen, right? So after addition of the antigen, initially when the antibody will be excess, significantly higher than antigen, then it will not be able to make this cross links, right? So that's why there will be very few antigen antibody complex, and rest of the antibodies will be free, right? So now in this case, this is antibody excess zone, so where antibody will be excess and antigen will be very low, so that's why they can't make the cross link and they can't make the precipitation. So initially there will be zero precipitation. Then when you keep adding the antigen, then this precipitation level will be increased, because uh, once you start adding the antigen, then you can see that some of them will make a cross link and it will give you precipitation. So gradually it will be increased upon addition of the antigen, and when it will reach to the equivalent zone, then here it will have a peak, because at the equivalent zone antibody concentration is equals to antigen concentration, right? So for that, as both of them of equimolar concentration, now this antibody and antigen will bind to each other and make this mesh like cross link, right, which will create a precipitation which is insoluble. So when your antibody concentration equal to antigen concentration, then you will get this equivalent zone and you will get a maximum precipitation. Again, when you keep adding the antigen, then now in this zone antigen will be excess but antibody will be at low amount, right, or limited. Antigen excess, antibody limited. So when antigen is excess, then again there will be lots of antigen, all of them will be so lots of antigen. So now all the antibodies are bound with the antigen, but there are some free antigen, right? So antibody excess zone, there will be some antigen antibody complex and some free antibody, but in antigen excess zone there will be antibody antigen complex and some free antigen, right? These are the difference, and as a result, when you cross the equivalent zone and if you keep adding the antigens, then the precipitation level of precipitation will start decreasing, right here. Why? Because as you increase the antigen concentration, now it is significantly higher than antibody concentration, so most of the antigen will be free, and some of them will bind to the antibody, and now it cannot make this cross link or mesh like network, so it cannot give you the precipitation. So when you cross the equivalent zone, the antibody precipitation level will be decreasing, right? So this is a typical bell curve for uh, this precipitation reaction. It has three zones: antibody excess zone, equivalent zone, and antigen excess zone. You will get the highest level of precipitation at equivalent zone, where antigen concentration equal to antibody concentration. So now if you centrifuge this antibody excess zone sample, then in the then of course some of the antigen will bind to the antibody and they will make an antigen antibody complex, and sometimes they can make this cross link to give you precipitation, right? So if you will centrifuge, then the precipitation will come to the pellet, and the unbound free antibodies will be in the supernatant. In the equivalence zone, if you centrifuge, then there is no free antibody or there is no free antigen. So you will get a precipitation which will be in the pellet, and in the supernatant there will be neither antigen nor antibody. Then when you will come to antigen excess zone, then there will be antigen antibody complex which will give you the precipitation here. So in the pellet you will have the antigen antibody complex, right? But in the supernatant you will have now excess antigen, so we will have the excess antigen or unbound antigen in the supernatant. So in the left thing, antibody excess zone, you will get uh, unbound antibody in the soup. In the equivalent zone there is neither antigen or nor antibody in the soup, and in antigen excess zone you will have uh, antigen unbound antigen in the supernatant. So this is your precipitation reaction, and it peaks at the equivalent zone. So the optimum condition for this precipitation, antigen antibody precipitation, is when both of them are in same concentration, and of course the antibody has to be polyclonal. So now next thing is diffusion. So all of these techniques uh, are based on this precipitation reaction, because here you can see that this precipitation is the okay. Am I audible? Can you hear me? Okay, so as you joined late, so already the discussion have been gone through this antigen antibody interaction, where we found the different kind of interaction between antigen and antibody here, which are hydrogen bond, ionic bond, hydrophobic, van der Waals interaction, and also we discussed the precipitation reaction, where we saw that the precipitation that which which is insoluble form, that can only be achieved when your antigen and antibody have the equal concentration, right? So this precipitation curve has three region: antibody excess zone, when there is excess antibody and you just start adding the antigen, then of course antibody is excess, antigen is limited. So as you increase the antigen concentration, the level of precipitation will be starting increased, right? And of course there will be some unbound or free antibody. When you reach the equivalent zone, there will be equal concentration of antigen and antibody, so it will give you the cross linking or the precipitation, right? And there is no free antibody or no free antigen. In antigen excess zone, when if you keep adding antigen up beyond this equivalent zone, then this antigen excess in this antigen excess zone, there will be some antibody antigen complex which will give you the precipitation, and rest are the free antigens or unbound antigens. So that is called antigen excess zone. And if you will centrifuge this antibody excess zone, then in the supernatant you will have the unbound antibody. In equivalent zone you will not get any antigen or antibody in the supernatant because there is no free antigen or antibody, and in the antigen excess zone you will get free antigen in the supernatant. So that's all we discussed till now. So now the several assays will be discussed which are based on the precipitation. Precipitation forms when antigen and antibody interacts and it will make a cross link, right? So that will give you a insoluble precipitation. That is the precipitation reaction, and which is the outcome of a antigen antibody interaction. So this thing will be measured to quantify the antigen or antibody level in the following assays. So first is immunodiffusion. So in immunodiffusion, uh, in a agar plate, most probably in 2% agar 2% agar plate, antigen is embedded in a well, right here. So now this antigen will be allowed to migrate through this agar, right, and this plate has a homogeneous concentration of antibody throughout the uh, plate, right. So now when antigen starts to be diffused through this agar, then there will be a point, a radius, where the antigen concentration will be equal to the antibody concentration, and it will give you the typical precipitation line, right? Because at this position, at this uh, periphery, you will have equal amount of antigen and antibody. That's why they are giving you the precipitation. This is your equivalent zone, right? So this will give you the precipitation. So here in so let's go by the so here actually you can see that this circle has lower radius, right? So this radius has accompanied with very low amount of antibody, so that's why here antigen is excess, antibody is limited, right? So all of the antibodies are bound with the antigen, but there are some free antigens, right, till this area. That's why it is an antigen excess zone. So right, so that's why you will have some free antigens and antigen antibody complex, but they are failing to give you the precipitation because the concentration, because there are some free antigens. So when it will go in this region, then now you can see the radius has been increased. So now as the radius has been increased, that means this periphery has also been increased, because the formula of the periphery is circumference is 2 pi r, right, which so if we go, so if r will be so if r will be increased, then this circumference will be increased. That means you will have the more antibodies here, right? Now when you get the more antibodies, then you can see then this amount of antibody will be equal to the antigen, right? So that's why as this has been turned into in a equivalent zone, so now you are getting a precipitation line here, right? So that's how you can measure that the concentration of the antigen which you embedded here. How? By plotting a graph. So basically you will you have to make a standard curve by taking some known concentration of the antigen, and you need to measure their migration where you are getting this precipitation line, right? So now let me just draw this here. Yeah, so you can plot a graph here. In y-axis you can get the distance, okay, in x-axis the antigen concentration, right? So you can plot a standard curve, and by different concentration of the antigen. So if you increase the concentration, the amount of migration will be more. So you will get a curve like this, right? This is your standard curve. Now you will add a antigen whose concentration is yet to be measured. You will add it into this well, and you will measure its distance of the migration where you are getting the precipitation line. Then you will measure this distance and you will plot here, and then you can find the corresponding concentration of the unknown antigen, right, by using this standard curve. So distance versus this concentration graph, that will give you the concentration of a antigen, right? So that is called radial immunodiffusion, and this precipitation line will be formed when the migration, the radius of migration will be such a will be in such a way so that the the amount of the antibody will be equal to the amount of the antigen. Then it will give you a precipitation line, right? So if your antigen has more concentration, it will migrate more, right? Because if it is more than this concentration, then it will require more antibodies to be enough to make this equivalent zone, right? If you increase the concentration to here, then it will need more antibodies, and more antibodies you can find at the further distance, right? Suppose, for example, if you are adding the antigen which is, suppose this is x concentration, right? So now if you are adding antigen of concentration 2x, then the migration will be more, right? So from here to here it will go till this point. Why? Because at this distance, if you will draw this circle, then this circumference can accompany that amount of antibody which is equal to 2x, right? So of course that will be farther than the x. So if you will increase the concentration of the antigen, then the precipitation line radius radius of that precipitation line will be increased. So that's how by from this plot you can get the concentration of a unknown antigen. Okay, any doubt here? Any doubt? Should I go ahead? Okay, so I think it is clear how will you able to measure the antigen concentration by the radial immunodiffusion, right? So another process is double immunodiffusion. Yeah. So again to mention, if you have any doubt at any place, so please feel free to stop me and ask the doubt. Okay. So in radial immunodiffusion, you can see that only antigen was migrating, and the plate was formed by with antibody, right? So in contrast, the double immunodiffusion, both antibody and antigen, they will move. Both of them will move. Okay. So this is your agar matrix. So the left side, in the well, antibody is provided, and in the right portion well, antigen is provided, and they were allowed to migrate through this agar matrix. So antibody will migrate from this well, antigen will migrate from this right well, and then so when they will meet here in equal concentration, right, then this meeting point, which contains equal concentration of antigen and antibody, in that region they will make a precipitation line, right? Because it will able to give you the cross linking and which will give you insoluble precipitation. So in this agar matrix, the position where you will get this precipitation line is the region where your antibody and antigen concentrations are equal. So in this technique, you can use this technique to measure concentration of antibody or antigen both, right? So this was immunodeficient. So now immunoelectrophoresis. Again, the uh, objective is same. For example, here to determine the concentration of the antigen or antibody. So here first, what is done? The first step is electrophoresis. You are given with the antigen sample, and now with the antigen sample, it can be a mixture of different proteins, right? So in the antigen sample or the antigen lysate, then you can get if it has different protein, then after electrophoresis you will get all the proteins separated, right? So first step is electrophoresis. Then you will add a antibody over over this gel here. Okay, so now these antigens which were separated by uh, I mean which were separated by the electrophoresis, now they will be allowed to migrate through the gel. Okay, so now there will be a point which is denoted by this blue dotted line, where now this antigen is migrating, antibody is also migrating in opposite direction. So when they will meet in the meeting point where the antigen and antibody concentration will be equal, right? For example, this yellow one, this yellow protein, when so it will start migrating towards the antibody, and antibody will start migrating towards the antigen, right? So now the position where this antigen and antibody concentration will be equal, there will get a precipitation curve, and that is true for the same thing will be uh, going on for each of the protein present in this antigenic samples. So first part is electrophoresis, where you will separate the proteins from your antigenic sample, and the second part is immunodiffusion, where they both of them will be antibody will be added, and this antigen and antibody will be allowed to be diffused through this agar matrix, and it will create a precipitation line where the concentration of the antibody and antigen will be equal. Now this distance will be measured, this distance where uh, distance from the distance from the antibody and to the precipitation curve. So this distance will be measured as the antigenic concentration of each of the proteins, right? So that's how we can measure the concentration of the antigen of each of the protein of this antigenic sample, right? By first electrophoresis and then immunodiffusion by adding antibody. So that's how immunoelectrophoresis works to measure concentration of. It is useful when your antigenic sample has more than one protein. It is a mixture of proteins. Then this technique will be very useful, because first you need to sort out, you need to separate those individual proteins, and for each of the individual proteins you can find this migration distance where it will give you the precipitation curve, right? So for each individual protein you will get a precipitation curve, and after that you can easily measure the distance, and you will plot the graph to measure the concentration of those individual proteins, which is the same way we discuss in the radial immunodiffusion, right? So that's how in immunoelectrophoresis you can actually measure the concentration of different protein of uh, antigenic sample which contains mixture of proteins. Then you will need this technique. Any doubt here? Any doubt in electrophoresis? Okay, so as there is no doubt in electrophoresis, so we can move ahead. Next is agglutination. So can anyone tell me what do you mean by this term agglutination? So agglutination is a term when the same we talked about equivalent zone where antigen antibody concentration is equal, then you will get a insoluble precipitation. Same thing happens when in a blood serum you add antigen, then if the antigenic concentration is equal to the antibody concentration, then it will create the precipitation which is known as agglutination. For example, here, so in this experiment, what has been done? They have taken the RBC. Yeah. So they have taken one particular protein from RBC cell, and they use the anti-RBC protein serum, and then they use the different concentration of the serum, they dilute it, and then you can see that each dilution is presenting the each different level of agglutination, right? Initially when the antibody was undiluted or less, or the dilution was low, then antibody has more concentration, so that's why it was able to make uh, lots of agglutination. But when you decrease the antibody concentration, then you can see the level of agglutination has been decreased here, right? Because this agglutination is the result of the cross-linking by antigen and antibody. So if you will be decrease the antibody, then you will get less available antibodies to make this interaction, so that they can make this antigen antibody precipitation. So that's why you are getting lower level of agglutination when you are diluting the antibody. Okay, so this the same concept has been used by the commercially available pregnancy test kits. So initially the pregnancy test kit, it contained two things. One is hapten carrier conjugate. So for the first of all, if a woman is pregnant, then her urine should contain the HCG hormone, right? But if the woman is not pregnant, then the urine will not contain the HCG hormone, right? So HCG will be present for a pregnant woman, it will be absent for the non-pregnant woman. So in the kit, you will be provided with a hapten carrier conjugate HCG. So this HCG, which is the antigen here, this HCG antigen is conjugated with a hapten carrier, right? And they will also provide the anti-HCG antibody, right? So now the test procedure is, you will take your urine and then anti-HCG antibody and this hapten carrier conjugate. So after the incubation, you will add the HCG carrier conjugate, then you will observe for the visible clumping. So the possible reaction is, so the two possible cases are, case 1, the woman is not pregnant, and case 2, the woman is pregnant. So just let me enter okay, so let's go back to yeah. So in order to decide whether the sample is pregnant or not, then then in case of negative, there will be no HCG hormone in the uh, in the urine, right? So upon the addition of the antibody and the HCG hapten carrier conjugated, they will make a cross link, which is a precipitation that will give you a visible clump. Why? Because the kit is already providing a hapten carrier conjugated HCG, right? When and the antibody, anti-HCG antibody. So when this anti-HCG antibody will react, will actually bind to this HCG, then they will make a cross link, right? Which will result into a visible clumping. So if there is clumping, so that means that sample does not have the HCG hormone, right? So now if uh, for the case of the positive case, if your sample contains HCG, then now your kit is giving you this hapten carrier conjugated HCG and antibody, right? So now in the presence of the HCG in the urine, this antibody will bind to this HCG in the urine sample, right? And then this antibody will entrap all the HCGs here, right? So now there will be no available antibodies which can bind to this hapten carrier conjugated HCG. So there will be no visible clumping. Again, in the negative case, there was no HCG hormone, right? So for that, the provided antibody was free to interact with this hapten carrier conjugated HCG, and it was able to make this cross linking, which will give you a visible clumping. But in the case of positive case, if your urine contains HCG, now this HCG will bind with the antibody which is provided by the kit, anti-HCG antibody. So now there is no antibody is available to cross link with this hapten carrier conjugated HCG, right? So in this case, you will not get any cross link, you will not get any visible clumping, right? So if you are not getting any clump, that means the sample is positive, and if you are getting a clumping, visible clumping, then the sample is negative. So again, the thing is about the competition. If there will be no available free antibody, then there will be no cross linking, no clumping. So the sample will be positive. And if there will be available antibodies when the sample will not contain the HCG in the urine, so now the free antibody, if there is the available free antibody, then they can be easily used to make this cross link with this hapten carrier conjugate. So in that case, you will get a clump, right? So that's how the pregnancy test kit works, by using this agglutination reaction between the anti-HCG antibody and HCG. So this was about the agglutination of antigen and antibody. So next we will shift to radioimmunoassay or RIA. So in radioimmunoassay, the idea is you can use a radio labeled uh, antigen, and you will have a plate or a well which is coated with the corresponding antibody, and then you will use the infected serum, or you can do the opposite thing also. You can uh, you can conjugate the well with antibody, right? And then you can use radio labeled antigen for that corresponding antibody, and also you can use the uninfected serum or infected serum for your test, right, which is not radio labeled. So again, what is happening in this technique? So this is your plate which is coated with anti HBs antigen, antibody specific for HBs antigen, right? And then when you will add the radio labeled HBs antigen, this red one, they will bind to the antibody and they will give you signal, right? Because they are radio labeled. But when you will use the infected serum, which are not radio labeled, now they will also give a competition to this radio label antigen, right? So some of the antibodies will be bound to the radio level antigen, and some of the antibodies will be bound to the unlabeled antigen, which is from the infected serum, right? So it will basically reduce the bound radio level antigens. In case of negative control, you will use an uninfected serum. Not negative control, if the serum is uninfected, then when you will add the uninfected serum as well as the radio level antigen, then uninfected serum does not have any antigen for this anti-HBs antigen, so it does not have any HBs antigen, so it will not bind to the antibody because it does not contain any HBs antigen, and this radio level HBs antigen will come and it will bind to the antibody, right? So in this case there will be no competition, so all of the antibodies will be bound with the radio level antigen, right? So it will give you very high, the maximum intensity of right, maximum radio intensity. But if you will compare this with this, in this case in infected serum, some of the antibodies will be bind with the infected serum which has the unlabeled HBs antigen. So if you will measure the intensity of this well, this intensity will be of course lower than this one, because here all of the antibodies are bound with the radio level antigen, but here some of the antibodies are bound with the radio level antigen and some of the antibodies are bound with unlabeled antigen, which is coming from the infected serum. So if we will plot a graph, if you increase the concentration of the unlabeled HBs antigen, means your serum which is infected, then the amount of radio level HBs antigen bind to the anti-HBs antigen will be decreased. So basically the radio intensity will be decreased if you increase the unlabeled HBs. If you will keep increasing the unlabeled HBs antigen, then it will keep overcompeting the radio level HBs antigen. So your unlabeled, so if you will increase the concentration of the unlabeled, then labeled one will be replaced and they will be exchanged with the unlabeled antigen. So the radio intensity will be decreased if you increase the unlabeled antigen, right? And in this case there will be no unlabeled antigen, so you will get a maximum intensity here. So if yeah, so in this case you will get maximum intensity, but in this case as you increase the concentration, intensity will be keep decreasing. Okay, so that's how this is a plot by which, so first you will plot a standard graph by measuring the intensity of some known concentration of the serum sample, right? So some known concentration of the serum sample you will put and you will measure the intensity, and for the new one, for the test one, you will measure the intensity and you will you can find the concentration by this standard curve. Okay, so in this way, by radioimmunoassay you can measure the concentration of the uh, antigen in serum, as well as you can also determine whether your serum is infected or not. If it is infected, then it will give intensity which is lower than the serum which is uninfected, because this uninfected serum will give you the intensity which is higher than the serum which is infected. So if your uh, intensity is lower than the uninfected serum, that means this sample is infected with NVS antigen, right? So two things: you can find whether the serum has HBs antigen or not, yes or no, or if it has, then what is the concentration, that you can make by this standard curve, right? So any doubt in this part, radioimmunoassay? So next is ELISA. ELISA is for again a way of measuring concentration of antigen or antibody. So there are three kinds of ELISA: indirect ELISA, sandwich ELISA, and competitive ELISA. In indirect ELISA, antigen well is coated with antigen, then after washing, to remove all the unbound antigen from the well, you will remove, and then you will add the specific primary antibody, which concentration has to be measured. So those antibodies will bind to the antigen, and upon washing, the unbound antibodies will be washed away. Then you will add enzyme conjugated secondary antibody. So this secondary antibody has an affinity towards the Fc region of the primary antibody. So they will bind to the Fc region of the primary antibody, and then again washing. So after washing, you can see that there is a enzyme which is conjugated with the secondary antibody. So for example, if the enzyme is HRP, then if you will give the substrate, then it will change its color, right? So now if you add the substrate, and you can measure the color intensity to which is actually proportional to the antibody concentration. In the same way, you can plot a standard curve where you will add known concentration of the antibody, and you will measure this color intensity, and then by the same way which I have shown you, that you will plot a graph. In y-axis you will measure the intensity, in x-axis you will measure the concentration of the antibody. So more the concentration, more the intensity, you will plot a standard curve. Then you will take an unknown sample, you will measure its intensity here, and then from this standard graph you can calculate its concentration by this. Okay, so that's how you can measure concentration of a unknown sample, unknown antibody, by using ELISA. Or you can use the different, and if you need to calculate, and if you need to know the binding affinity of different antibodies for a particular same antigen. Suppose you are given with the same antigen, and you are given with some different antibodies from different patient serum, and you are asked to check out which antibody is the best suited, which has the maximum binding affinity. So for that you will yeah, so for that you will take different antigen, different serum, and then you will plot this graph by measuring the intensity of the light of which is from this reaction of substrate. So in for example, not this graph, you will get a bar graph. Suppose you have given with antibody 1, antibody 2, antibody 3, and you are asked to check which one is the best for a particular antigen. So you get 3 bar for antibody 1 and 2. This is your antibody 1, this is antibody 2, this is your antibody 3, right? In y-axis there is intensity, which is actually indicating the affinity. So you can see that the antibody 2, this antibody 2 or the serum 2, it has the maximum intensity of this with this antigen. So your answer will be antigen 2 is the best. Antibody 2 is the best. Next is sandwich ELISA. So in sandwich ELISA, the well is coated with the antibody. Washing is done to remove the unbound antibodies, and then you will add the antigen over the well. So the antigens will be bound to the antibody which are coated to the well. and then after washing you will add the secondary antibody. Now this time difference is here secondary antibody was binding to the Fc region of the primary antibody, but in this case sandwich ELISA the secondary antibody will be bound to the antigen which is already bound with the another antibody, primary antibody, right? So here this secondary antibody will be bound to the antigen which is bound to the primary antibody. Then again you will add the substrate and you will measure the color intensity, right? So to know the concentration of the antigen which is added after antibody coat. Next is competitive ELISA. So the next is competitive ELISA, where you will incubate the antibody with the antigen to be measured. So this is your antibody, and you will add the antigen whose concentration has to be measured, right? So now we will mix this, we will add this mixture to the well, right? So now this well is coated with the antigen, right? So now when you add this thing to this well, then the free antibody will bind to this antigen which are coated into the well, and the antibody which was bound with the previous antigen, that will be unbound, right? So that will fail to interact with the antigen which is coated. So this antibody will not be coated. So upon washing, this antibody will be removed from this reaction mixture. Then you will again add the secondary antibody and you will check the color intensity. Then you will know that the intensity, the concentration of this antigen. How? By this way. Suppose initially you have taken lower concentration of this antigen. So in this case, availability of this free antibody will be more, right? So if there is more free available antibody, then you will get more signal. Why? Because more of this antibody will bind to the antigen coated with the well, and this antigen is different, right? So okay, so in this case you will get more signal because the antibody which were bound previously with this antigen, they will be washed away. So only the antibody which is free, and that can only bind to the antigen which is coated here. So only this antibody will be stayed, this bound antibody will be removed by washing. So if you have the very less antigen concentration initially, then you will have very high amount of signal, because less antigen means more free antibody means more binding and more color intensity. So you will get the very high signal at when your antigen level is very low, right? For example here, now if you will increase the antigen concentration, then what will happen? Availability of the free antibodies will be decreased, right? So now if the free antibody will be decreased, then you will get lesser amount of signal, because the free antibodies are the antibody which will bind to the coated antigen and they will give you a signal. But in this case, if your maximum antibodies are occupied, then they will fail to interact with the coated antigen, so the signal will be reduced. So as you increase the antigen concentration here in this antigen, then you will lose the color intensity. So the graph will be like this. If you will increase the antigen concentration, the intensity will be decreased, right? So this is your standard curve. And now if you have a known unknown antigen, then you will set up the same reaction and then you will measure this intensity. If it falls like this, then you can find its concentration from this standard curve, right? So that is your competitive ELISA, where you are competing. This antigen will bind to the antibody, and uh, only the free antibodies will be the one which can interact with the coated antigen, and they will give you signal, right? So this is your competitive ELISA. So indirect ELISA, sandwich ELISA, and competitive ELISA. All of the three are very important, and they have different purpose of doing. So the difference of sandwich and indirect ELISA is, here the secondary antibody in the sandwich ELISA, it will bind to the antigen, but in indirect ELISA, the secondary antibody will bind to the Fc region of the antibody, right? So this is the difference, and that's how, by plotting the standard curve, you can measure the concentration of the antigen or antibody, whatever you are doing, uh, by those three experiments. Okay, any doubt here? So the next is ELISpot assay. So in ELISpot assay, you can locate the position of a cell which is secreted some cytokines, right? So here the motto is to find out the location of the cell which are secreting some, for example, cytokines. So here in this ELISA well plate, you will coat the well with anti-cytokine antibodies. Then you will add some the cell mixture which contains the S or cells which can secrete, and NS non secreted cells which cannot secrete. So now you will add both S and NS. You will incubate at 37 degrees Celsius, and then you will, after washing, you will discard the unbound cells. So your secreted cell that will secrete cytokines. So the cytokines will bind to this anti-cytokine antibodies which is coated to the well, right? And your NS, the cells which cannot secrete, they will not secrete any cytokine, right? So after this washing, the cells which are not attached to this antibody, they will be released. Only the antigen which was secreted by the S cell, that will be stayed here, right, as they are bound with the antibody. Now we will add the secondary antibody which is enzyme linked, and then when you will add the chromogenic substrate CS, then it will be changed into the colored product CP. Then you will see a blue color here, only at the positions where the cells they were able to secrete cytokines, that particular cytokines. On right, so on position of the location of only those cells will give you this spot, but the location of the non-secretory cell, as they have not secreted anything, so so this there was no cytokine which was bind to the antibody, right? So that's why you will not see any blue color here. So this ELISpot assay is for locating the site of secreting cells who can secrete cytokines, right? Next is Western blotting, which is very simple. Simple. You will prepare your cell lysate, and then you will treat them with SDS to denature the cells and to impart a negative charge, and then you will do the electrophoresis from a negative to positive, because your proteins are now negatively charged, so they will be migrated from minus to plus end, and then you will transfer the proteins from this gel to PVDF membrane, and then you will process the membrane with primary antibody and the secondary antibody, if you, and then you will add the substrate, and then you will see that whether you are if the enzyme is converting a substrate to a colored product, then you will see the color, and you can see that yes, the in this lane the protein is present. So that is your Western blotting. And then immunoprecipitation is also an example of technique where you can purify proteins by using antigen antibody interaction. So in the test tube, see, in the test tube you will have the cell lysate, and then you will add the specific antibody. The specific antibody which is specific for this red protein, it will bind to this red protein, and this antibody are after that you will add the secondary antibody. The secondary antibody will be bound to the primary antibody, which are bound with their antigen, right? And the secondary antibodies are coupled with the magnetic beads. So now so after that you can apply the magnets and the rings to remove the unbound material. So you will be only left out with the antigens which are bound to this antibody, right? So that's how you can only purify or precipitate this red proteins from this entire cell lysate. This is called immunoprecipitation. And flow cytometry for so for this, suppose you have a population of you have a cell mixture where you have mixture of different population of cells. Some of them are expressing protein A in their surface, some of them are expressing protein B, some of them are expressing both of them, some of them are expressing none of them. So now we will add anti A and anti B antibody. So so they will bind to the corresponding site, right? Now after that you will pass this cell mixture through this ultrasonic nozzle vibrator. Then you will get the droplet of the cells, and which are actually charged, ionized. So now when you will inject a laser beam towards these droplets which are coming from this nozzle, so after this laser beam injection, this deflection plate, they have minus and plus charge, so they will decide at which direction you should go. So there are three possible cases. One is the cells which has A minus B positive cells and A plus and B negative cells. Second mixture will contain A minus B minus cell. Third mixture will contain A plus and B plus cell. Now we can process the data in the software, and you can plot a graph where in x-axis we can plot the anti-A antibody fluorescence, y-axis anti-B antibody fluorescence. So if you will divide the segment with 4 quadrant, then in this one, in third quadrant, there will be the cells which have anti A minus B minus. That means it is neither expressing A protein nor expressing B protein. So it will not give you any signal. For example, this first quadrant, A minus B positive cells, so this is bound with the anti-B antibody, and this anti-B antibody is tagged with some fluorescence, is conjugated conjugated with some fluorescence, for example green fluorescence, then you will see a green I mean you, you can see all those cells in that green channel, right? And the cells which are producing protein A but not protein B, then those cells will be bound with the only anti-A antibody, right? So in that case, if the anti-A antibody is conjugated with uh, fluorescence red, then in the red channel you will get these cells, right? And then if you merge this, green and red, then you will get the cells where you will find both uh, the cells which are expressing both A and B. That's why they are bound with both anti-A and anti-B, right? That means they will emit the red as well as green. So they will be in the second quadrant. So that is the flow cytometry, where we can sort the cells based on the protein they are expressing in the cell surface. And then northern blot is for analyzing the RNA. So from your sample you will isolate the RNA, you will do the electrophoresis, then you will transfer the RNA to the PVDF to the membrane, and in the membrane you will hybridize the RNA if there is any secondary structure. Then you will add the probe which is radio level, then you are looking, and if you are looking for a specific RNA sequence, then you will design the probe in such a way that the probe will contain that actually complement to that particular specific RNA sequence. So only those region that particular probe will go and bind, and then as the probes are radio level, then you can see those probes which are bound with the specific RNA sample RNA molecule here. Those RNA molecules can be visualized by the autoradiography because it is bound with the probe which is radio level, right? So that's how we can do northern blot to visualize your specific RNA molecule. So now we can go to the question answers session. So the first question is, precipitation reaction carries out in agar gels are referred to as agglutination, immunodiffusion, flocculation, or neutralization. We just read that precipitation when occurs, it is called as immunodiffusion. See, when it will diffuse, the antigen was allowed to diffuse, then it was found the area where it get the antibody of equal concentration, and right, so in that region was the part of the equivalent zone, and it created a precipitation line. So the answer of this question will be, precipitation reaction occurs in agar gel that is referred as immunodiffusion. So option B is correct here. So next is, the result of ELISA test is determined by measuring the intensity of the color, intensity of the radioactivity, change in chemiluminescence, diameter of the precipitant ring. Precipitant ring diameter is measured for this immunodiffusion assay, right? For radial immunodiffusion, if you remember here, this diameter is measured, or the radius, whatever you say, this radius or diameter is measured. More the distance, more the concentration. If you will increase the concentration of the antigen, then the distance will be more, the diameter will be more, right? So here the concentration is measured by the diameter of the precipitate ring, right? So of course this cannot be the answer here, because in ELISA test we don't measure the diameter. Change in chemiluminescence, no, that is for Western blotting, where the secondary antibody is linked with the substrate, and then when the secondary antibody is linked with the enzyme, then when we provide the substrate and the corresponding enhancer of the chemiluminescence or ECL, then you will get a chemiluminescence. So this is for the Western blot, not for ELISA. Intensity of the radioactivity, no. Radioactivity we measure when for southern blot, for northern blot, but not for ELISA. For ELISA we measure the intensity of the color. Right? See here, the enzyme is linked with the secondary antibody, and we use a chromogenic substrate which is converted into a colored product. Right? So this color will be measured, intensity of the color will be measured to know the concentration of the corresponding protein. Right? So here the answer will be option A, intensity of the color. Incubation of the microtiter plate with a non-specific protein solution after coating of antigen or antibody is known as. So basically for ELISA also you coat your wells with antigen or antibody, and then you incubate the antigen or antibody coated well with a non-specific protein solution, for example BSA, right? BSA which has the ability to block the empty space of the wells, because after coating the wells with antigen there will be some empty spaces, right? So now after that when you will add the antibody, then antibody can go and bind into the empty spaces of the well, right? So that can cause a false signal for you. So that's why you need to block this region by non-specific protein solution, for example BSA, right? So this process is called as blocking. So answer is blocking. Now next is what is the purpose of adding secondary antibody while performing Western blotting or ELISA? In both Western or ELISA we add the secondary antibody, but they are asking for why. Option A, block any interfering noise coming from membrane or well. No, we are not blocking any well. We are not blocking any noise. That we do by in a different way, right? We use BSA for that, for blocking any empty space in the well, so that you will not get any non-specific signal. Option B, for ensuring proper binding of primary antibody to membrane or well. No, that binding of antibody to well is not depending on secondary antibody, right? It is depending on the antigen which is coated to the well. So option B is also false. Allow detection of antigen sample by binding primary antibody. Yes, that is true. Detection of antigen sample by binding, because secondary antibody binds with the FC region of the primary antibody, right? Which in turn give you the it change the color, or it will show the chemiluminescence to allow the detection of the antigen. So it binds the primary antibody to show the detection of the antigen sample. So option C is right here. The correct sequence of the Western blotting is first performing page, second transfer protein to the PPDF membrane, third protein denaturation by SDS and by boiling, incubate the PPDF membrane to primary antibodies and secondary antibodies, right? So the first step is the sample preparation, where proteins will be denatured by SDS and by boiling, because proteins should not be in a native state and proteins has to be negatively charged. So this is the first step, option 3. Then we will perform the page electrophoresis. So then one will come, and then we will transfer protein. After the electrophoresis we will transfer the proteins to the PPDF membrane. So this option, so then come the 2, and then at last we incubate the PPDF membrane to primary antibody and secondary antibodies subsequently. So option C is the correct sequence for Western blotting. In the immunostaining technique, the proteins are stained with antigen, Kumasi blue, silver stain, antibody conjugate to the fluorophore. So for immunostaining, as you know that for immunostaining, if this is a protein which you need to visualize, then you will use a primary antibody which will bind to this protein, and then you will use the secondary antibody which will bind to the Fc region of the primary antibody and which is conjugated with some fluorophore probe, for example FITC or DS red, right? Then it will give you a, if it is conjugated with FITC fluorophore probe, then it will give you a green fluorescence upon exciting with a proper wavelength, right? So the answer will be antibody conjugated. So if you will see the green light, that means you are visualizing this protein. So in this immunostudy technique, the proteins are stained with the antibody conjugated to fluorophore. Option D is correct here. To make the intracellular target accessible to the antibodies, permeabilization is performed. All are permeable, all are the permeable agents except Tritonex, Twin 20, saponin, paraformaldehyde, right? Tritonex 100. See, anything in this question, anything detergent you will find, that will be the permeable agent. Why? Because the job of this non-ionic detergents, for example Twin 20 or for Tritonex 100, they are, all of them are non-ionic. So what they will do, they are not that strong, so they will solubilize the membrane. So right, that is the job of detergent. So they will make a small holes in the cell membrane, right? So they will not completely denature, or they will not completely take out the membrane, but they will make a small pore in the membrane, right? Because they can solubilize the membrane. So that's why this Triton X100, it is our permeable agent. Twin 20 is a permeable agent. Saponin is a permeable agent, but paraformaldehyde is not a permeable agent. This is not used for, you know, permeabilization of the antibody. It is used for the cell fixation, so that the cells can be fixed, right? So option D is right here. Which one of the following statement is a true for flow cytometry? Forward light scatter is related to the size of the cell. Forward light scatter is related to the granularity of the cell. Side scatter is related to the size of the cell. Side scatter is related to the granularity of the cell. So forward light scatter FSC is always related to the size of the cell, right? For example, if you plot this graph, if you plot the FSC here, right, and if you have multiple samples, then if your cell size is more, then the FSC will plot your largest cell at the right here. Largest cell as the, sorry, if you will plot this graph in such a way that here is size, right, size, and here is your FSC, then FSC will plot the largest cell to so it will give you the highest FSC value, because it can scatter light the most. More the size of the cell, more forward scattering. So if it is, for example, if the size is very big, so it will give you very high FSC. If the size is low, then it will give a lower FSC, right? So option 1 is right. FSC is related to the size of the cell, and so FSC, so option 2 is false. Side scattering is related to the granularity of the cell. If your cell is this, if this cell is opaque, there is no such granules, then it cannot create the side scattering. But if the cell, for example neutrophil, if it is completely granular, right, for example cytotoxic T cell, they are very much granular, so they can do the light side scattering very well, right? So for the side scattering, if you will granularity and side scattering, then the cell which is more granular, that will show more side scattering. The cell which is not granular, that will show lower side scattering. So that's why option 3 is false, but option 4 is right. So that's why option C is right. That FSC is related to the size of the cell, and SSC is related to the granularity of the cell. Next is the technique is that uses the laser to analyze the characteristics of the cell. ELISA, no, ELISA does not use laser. It uses the primary and secondary antibody for the concentration. Western blot, it doesn't use laser. Flow cytometry, yes, it does. It does use laser to analyze the feature of the cell, right? Which kind of receptors they are expressing. That characteristics can be found by fax, and this flow cytometry can use the laser beam, right? So for the question 9, option C is the right answer. Also, immunodiffusion is, it doesn't use the laser. Next is the increase of the cell surface MHC class 2 expression of activated macrophage can be visualized by ELISA, Western blot, immunodiffusion, immunofluorescence. Right. For ELISA will not let you the visualization of the MHC2 protein, right? It can, okay, anyway, it can, if you will use the antibody, then it can help you to quantify the concentration of the MHC class 2 expression, but it will not let you to visualize. Similarly, Western blot is for protein detection, so it will not you I mean, you cannot visualize by Western blot. Immunodiffusion also not. It is for calculation of the antigen or whatever protein you are taking to quantify the concentration. Immunofluorescence will help you. Suppose, for example, this cell, for example B cell, it is expressing the MHC class 2, okay, class 2 expression here. Then you can use a primary antibody against this MHC class 2 protein, right? And then you can use a secondary antibody which will use the which is conjugated with a fluorophore, for example DS red. Then you can, if you will excite your sample with a proper wavelength, then you will see a red color here. This red color will dictate you the location or the localization of the MHC class 2 protein. So the answer is immunofluorescence. Okay, so I think so till now we are done with the session. So if you have any question, please feel free to ask, otherwise we can close the session. Any doubt you have? Okay, so I think we can close the session. Okay, so see you on the next week.